All right, grade 12s, here we are back again. We're going to finish our chapter 19 on linked genes and just kind of picking up where I left off. We were discussing pedigrees that were trying to show a certain inheritance pattern like autosomal dominant or sex-linked dominant, sex-linked recessive, autosomal recessive. And I was trying to muddle through this and I thought maybe this could be sex-linked dominant. And you can see, and then I was trying to experiment with what the possible genotypes were. And I kind of just from trying to make things work, I noticed that it could not work as sex-linked dominant when I got to this male right here, and kind of understood that if it was dominant, this daughter would definitely be affected. So again, sometimes you experiment with the genotypes to see what would work. So this would only work as autosomal dominant. Then it works just fine in that situation, because we can see that the trait is every affected individual has an affected parent. We're not skipping a generation. And notice here, when there's no dominant allele at all, then we don't see it showing up in the offspring. So I just wanted to clarify that from last video before we move on to finish off linked genes. There is one more example here, which is a sex-linked dominant situation. So this actually works as sex-linked dominant. Again, works well for dominance because we don't skip generations and every affected individual has an affected parent. And this time, if we were to assign X's and Y's, we would see that sure enough, see if we focus on this male right here, this male has the dominant allele on the X, so every daughter must be affected, must. And however, the sons are fine because they're getting the Y from this father, you see? So no problems there. And up here, if it's dominant, this female is just a carrier. And so therefore there's sort of this 50-50 chance for offspring, because a boy could get the affected X or the normal X. 50-50 opportunity there. And then we see here that this female got that X. So this works as sex-linked dominant. Now, to finish off chapter 19, we need to just skip ahead here a little bit and talk a little more about Thomas Morgan. So not only did he determine that there are genes that are found on the sex chromosomes, he also did experiments again with Drosophila where he would look at two traits. And when he did experiments, he did not see certain expectations in the phenotypes of the offspring. So let's look at exactly what he did here. So let's say we're going to work with two traits of a fruit fly, body color and wing shape. Body color we're going to assign A, and the wild type is dominant. And then the black body color, which is recessive, we'll get the little a. Big B for straight wings, that's dominant and typical. But the mutated allele, the little b, is for curved wings. So we're essentially doing a dihybrid cross here. So Morgan takes pure breeding parents for both the dominant trait. So we have our capital A, capital A, capital B, crossed with our double recessive. So pure breeding parents for those traits. Lo and behold, if we do this cross, in the F1 generation, you can tell that that's the only possible genotype is big A, big B, little b, which of course means all the F1 offspring have the wild type body and the straight wings. And that's completely expected and normal. So of course, what are we going to do? We're going to do an F1 cross to see what our F2 generation would be. This would be a typical 4x4 Punnett square for a dihybrid cross. We're not going to take the time to fill it in. We certainly could, but you know what we would be expecting. So this next slide shows us what we would normally be expecting in the F2 generation. We'd be expecting this 9-3-3-1 ratio. 9 individuals with both dominant traits, 3 where one of the traits is dominant and the other trait is the recessive trait, and then 3 where we'll switch that around, and then 1 with only both recessive traits. However, that is not what was discovered by Morgan. He pretty much saw a 3/4 wild type straight, which is both dominant traits, and 1/4 of the offspring that had black and curved wings, the recessive, double recessive. And so he asks himself, well, and he did actually see a very small number of sort of where one is the dominant trait as one is the recessive trait. Very, very small numbers there. Not the typical 9-3-3-1. So he kind of asks himself, well, what's going on there? Well, what he proposed was that the gene for the body color and gene for wing shape were located on the very same chromosome, that they were actually linked. So that was not kind of following our typical Mendelian situation where we assume that the genes are independent from each other, which means they would be on completely separate chromosomes and they're able to align during meiosis independent of each other. But this time, if they're linked, they are dependent on each other. So he was right. So again, if we look at this, if we look at what's happening, we can see that this is what we should see in the gametes, but now we're only going to be looking at one chromosome. So the B and the A allele are found on the same chromosome. So for this parent, we always have a capital A with a capital B. For this parent, we always have a little a with a little b. And we find that in pretty much equal ratio, right? When we get our gametes. So those are the only possibilities of the gametes. And of course, that created our first F1 generation just fine. That would be pretty typical. So only two possible gametes is what we see for the F1. But then, when we do the F2, we know that remember we're working with an big A, big B, little b, crossed with big A, big B, little b, so the heterozygous parents this time. And if they're linked, we don't get 4 possibilities, we only get 2 possibilities for the gametes from each parent, because the dominant alleles stay together and the recessive alleles stay together. So this kind of explains why we saw a 3-to-1 ratio. Remember, I showed you that Morgan found that 3/4 pretty much had the wild type body, straight wings, the double dominant, and 1/4 had the double recessive. However, he also observed that very small percentage of the one dominant trait, one recessive trait. So what was going on there? Well, we kind of know what's going on there because of our study of meiosis, right? How can we get some more genetic variability in formation of the gametes? We get that when there's potential crossover happening. Remember where we get the intertwining of chromatids, and we can actually get a section of DNA that can trade places. So if that happens, that's going to allow for some variation in the gametes, where maybe a dominant capital A does connect to the recessive little b allele. And we're going to see that in a second, all right? So the only way that can be explained, as we can see, I'll just fast forward this here, is crossing over. So these are the dominant gametes, right? Where the genes are linked and it's the expected double recessive, double dominant. But when crossover happens, remember crossover is a random event, we can actually get a segment that trades places, which could bring together a dominant allele with a recessive allele, but only in small numbers. So that's, and when that happens, we call those gametes recombinant gametes, okay? Because we've recombined the alleles, the way that the alleles can come together. Okay? We also call individuals that don't look like one of the parents, we call those individuals recombinants, because they have one trait of one parent matched with another trait of the other parent. Not all looking like one of the parents. Okay, so I hope that made sense. That explains that very small ratio or frequency of recombinant individuals, individuals that don't look like one of the parents. Remember, the parents were all had both dominant traits or both recessive traits, and that was in the parent generation or the F1 generation. It didn't matter. It was in the F2 generation that we saw the small percentage of one dominant trait, one recessive trait. Well, that becomes very useful. It's not just useful to know that we can have genes found on the same chromosome. We can actually use that frequency of recombinance to know how far apart those genes are on the chromosome. So let's see how that works. And that ends our chapter. So we're going to use the frequency of crossover. So how do we get this frequency of crossover? We have a little formula, which you usually don't have to do very often. Usually you're given the results, but let's see where that crossover frequency comes from. So we would simply take the number of recombinants, remember those are the offspring that don't look like the parents, and divide that by the total number of offspring, and since this will always be a decimal, we multiply by 100 to get a percent. So let's say these were the actual results from the cross, where we were looking at body color and shape of the wings. And remember that we get approximately a 3-to-1 ratio of the double dominant trait to the double recessive, but then we saw small numbers of the recombinants where they had the one dominant trait and the one recessive trait. So you can see here that we have 18 total recombinants. If we added up the total number of offspring, we would get 400. So in the next slide, we actually do the math, and we end up with an answer of 4.5%. Well, that is an indication of how far away those genes are on the same chromosome. The greater the percentage of recombinants, or the greater the crossover frequency, the farther apart those genes are on the chromosome. Because crossover is more likely to happen when the genes are farther apart, right? That kind of makes sense, right? If the genes are really close together, it would be hard to split them. Much easier when they're much farther apart. So that's a good thing to remember. It's a good relationship for you to know. So this value, this crossover percent, we actually use it to map where the genes are on a chromosome. So it's a bit of a puzzle. The way we do this, the simple rules are, first of all, we need the crossover percentages, and we realize that actually can be converted to a distance. The distance is a relative distance. We're not using like nanometers or micrometers or anything like that. We use what are called map units. So it's a relative term that's used for distance on a chromosome. And everything has to add up. It's a bit of a math puzzle. The best thing to do is to look at an example. The big strategy is to start with the genes that appear to be the farthest apart. Again, this makes sense when we look at an example. So let's look at this gene mapping example. So let's say this time we're going to be looking at 3 genes. So 3 linked genes all on the same chromosome. And let's say we've done the experimentation where we've done the pure crosses, we've done the F1 crosses, and we've used the results and that crossover percentage formula to actually calculate some percentages. So let's say we've done all the work and we find that body color gene or allele, little b say, compared to wing size, VG, we'll use that symbol, we get a crossover percentage of 17%. Well, we convert that into map units. We do the same thing with body color and the third gene, eye color, and we find that the percentage of recombinants in the offspring is 8%. That becomes 8 map units, and then we do the same thing. We need to have enough information to be able to plot basically a number line for where these genes exist on the same chromosome. Here's the kicker. Where do you start? Start with the genes that are farthest apart, which is, of course, your greatest crossover percentage or frequency, the most map units. So we're going to start with the 17 map unit distance apart of body color and wing size. So what we would typically do, it's like creating a number line, we would put 1, and it doesn't matter if we would have body color here or wing size. We just know that 17 map units apart is where they need to be. So we've got our wing size here and our body color, and we know that that's 17 map units. So where are we going to put the third one, the eye color allele or gene? Well, we know that it's 8 map units away from eye color, or I'm sorry, away from body color. So here's our body color, so we know that 8 map units away is where eye color should be. And we are assuming that eye color is 8 map units this way, to the right, because we've started with the 2 genes we believe are the furthest apart and that the other genes will fit in between. Now you have to be careful with that, because I have seen some questions where if you made that assumption, you might be in a little bit of trouble. In other words, they didn't give you the greatest number between the 2, because sometimes you can still do an effective mapping without seeing the 2 genes that are the farthest apart. That's when you get more alleles to put on the same chromosome. So what I mean is that the eye color could have been 8 map units to the left of body color. It could have been based on that one piece of information, but we have to use all the information. If we use the last piece of information, is that the eye color gene is 9 map units from wing size. So of course, if we put our eye color 8 from body color, then it makes mathematical sense to put the wing size 9 units this way, right? And so mathematically, it's like a number line, and it works. That gives us the relative distance apart of these genes on the chromosome. Now, the only way this gets a little harder, of course, is if things get a little bigger. So let's look at an example where we have 4 genes or alleles, and we're just going to give them letters of the alphabet, A, B, C, D. So we have to put these alleles, given the information that's there, on a chromosome. So it is like making a number line. So this is going to represent my chromosome, and I have to put my letters. Now, where do we start? Greatest crossover frequency, 45% for alleles B and C. So guys, remember, that is equal to 45 map units, and I'm going to put my B here, assuming that these alleles are forming the ends of my sequence. Therefore, C would be there. Now my next thing, let's just work with A. So where's A going to be? So it tells me that A is 30 map units away from C. So that's either 30 map units this way or 30 map units the other way. But I'm going to be reasonably assured that it's going to be found between my 2 extremes. Remember, it has to work out mathematically, so you can make an assumption, but it just won't work, and you might have to change it as you go. That's why we call it sort of like a puzzle. So we're putting A. So remember that this grand total here is 45. So we're assuming, so let's just say there, we can put A, and so we're thinking that is 30. Well, we only have B to put in. Now, B is supposed to be 40 map units away from D, right? 40 map units. So there's my B, so we're gonna come this way with D, 40. Well, gosh, that would put it pretty close to C, right? Because it's only 5 more map units to get to C. So this, I'll change the color here, this distance right here is 40. Now, if we've done this correctly, when we check our last percentage frequency, it should make sense. It should look right. So D from A is 25 map units. So here's A to D. So remember, A to C is 30. A to D is supposed to be 25. So if we've done this correctly, there's the 25. We can see that D to C must be 5. Now we don't have that piece of information. We don't need that piece of information, but it's making sense. It actually works mathematically. So our sequence is B, A, D, C, or C, D, A, B, because it really doesn't matter if you go backwards. Sometimes that's a very common perhaps numerical response question or multiple choice question where they just randomize the sequence. In other words, it's more about the sequence of the letters than actually worrying too much about how far apart they are. So let's just make sure our slideshow is done. So there will be practice questions for you to try with gene mapping, and hopefully you understood how we were able to prove that genes can be linked on the same chromosome or they can be found on a sex chromosome.